Hi everyone, my seminar topic is ribosomes. Here we are going to discuss about the ribosome structure, chemical composition and reconstitution experiment of ribosomes. Ribosomes are small, dense, rounded and granular particles of ribonucleoprotein. They serve as the factories of protein production. With the exception of mature mammalian erythrocytes, ribosomes are found in all prokaryotic and eukaryotic cells. In prokaryotes, ribosomes are freely distributed in the cytoplasm, and in eukaryotes, they are found freely dispersed in cytoplasm. Few of them are seen inside the mitochondria and chloroplast, and others remain attached to the endoplasmic reticulum outer nuclear membrane. There are two forms of ribosomes, monosomes and polysomes. Monosomes are protein synthetically inactive solitary ribosomes, and the polysomes are protein synthetically active functional groups of ribosomes. So, in the structure of polysomes, there will be an mRNA and several ribosomes attached to the mRNA like the beads on a string. This is a type of ribosomes. And according to the size and the sedimentation coefficient, there will be two types of ribosomes, 17S and 18S ribosomes. 17S ribosomes is found in prokaryotes and the 18S ribosome is found in eukaryotes. 17S ribosomes is smaller in size and have a sedimentation coefficient of 17S, and they occur in the prokaryotic cells of the blue-green algae and in the mitochondria and the chloroplast of eukaryotic cells. And the 18S ribosomes have the sedimentation coefficient of 18S and these occur in the eukaryotic cells of plants and animals. Next is the structure of ribosomes. Ribosomes are spheroid structures of 115 to 250 angstrom in diameter, and each ribosome is porous, hydrated, and is composed of two subunits, a larger and a smaller one. The larger subunit is dome-shaped and smaller one is placed above the larger one, and it is just like a cap in a cup and cap arrangement. In between the two subunits is a groove in which the mRNA is positioned during the protein synthesis. The larger subunit has a central canal in which the nascent polypeptide chain undergo elongation during the protein synthesis. 17S ribosomes has a larger subunit 15S and a smaller subunit 13S, and the 18S ribosome has a larger one 16S and the smaller one 14S ribosomal subunit. The two ribosomal units remain united with each other due to the high concentration of magnesium ions. When the concentration of magnesium ion reduces in the matrix, both the ribosomal subunits get separated, and usually they are found to occur freely in the cytoplasm and they unite during the process of protein synthesis. At high concentration of magnesium ions in the matrix, two ribosomes become associated with each other and known as the dimer. And during the protein synthesis, many ribosomes are aggregated due to the common messenger RNA and form the structure polyribosomes or the polysomes. These are all about the structure of the ribosome. Next is the models of ribosome structure. First one is quasi-symmetrical model. This model is proposed by Stoffler and Wickman in 1977. According to this model, the 13 ribosomal subunit, that is a smaller subunit, is elongated, slightly bent prolyte shape. It is a bipartite structure. In transverse hollow or the cleft, divide the 13S subunit into two parts, the smaller head and the larger body. 15S subunit is bilateral symmetrical and shows three protuberance arising from a rounded base, the central protuberance being the most prominent. The 15S subunit can be compared with the armchair. Its rounded base forms the vaulted seat and the central protuberance forms the back, and the lateral protuberance is compared with the arm of the chair. The 13S and the 15S subunit become associated to form the 17S ribosome. The long axis of 13S subunit is oriented transversely to the central protuberance of the 15S subunit. A tunnel is formed between the hollow of the smaller subunit and the vaulted seat of the larger subunit. through which the mRNA is passed during the protein synthesis. Next is the Lake's model. This model was suggested by James A. Lake in 1981. According to this model, both the subunits are asymmetrical. The smaller subunit has three parts: the head, base, and the platform. The platform separates the head from the base with the help of a cleft, and this cleft is an important function region. It is a site of codon-anticodon interaction and binding site for the initiation factors. In the larger subunit, we can see the ridge, central protuberance, and a stalk, and the ridge is separated from the central protuberance by the valley. The smaller subunit is positioned over the larger one in such a way that its head is aligned to the central projection of the larger one. So, a groove is left between them for the orientation of mRNA. Next is a 3D model of 18S ribosome. In morphology, this 18S ribosome is similar to the prokaryotic ribosome. The 14S ribosomal subunit in the 18S ribosome is divided into two, the head and the base, by a transverse groove. And the 16S ribosomal subunit is generally rounder in shape than the smaller subunit, and its one side is flattened, and this is the side that becomes confluent with the small subunit during the formation of monomer or monosomes. Next is the chemical composition of ribosomes. The ribosomes are mainly composed of RNA, proteins, and the metallic ion. Lipid and carbohydrate is absent in ribosomes. The 17S ribosome contains more RNA than the protein, and the 18S ribosome contains less RNA than the protein. First is ribosomal RNA. In 17S ribosomes, there are three types of ribosomal RNA: 23S, 16S, and 5S. 23S and 5S ribosomal RNA is found in 15S ribosomal subunit, and 16S ribosomal RNA is found in 13S subunit. 23 ribosomal RNA contain 3300 nucleotides, and 5S ribosomal RNA contain 120 nucleotides, and 16S ribosomal RNA contain 1615 nucleotides. In 18S ribosomes, there are four types of ribosomal RNA: 28S, 18S, 5S, and 5.8S. 28S, 5S, and 5.8S ribosomal RNA are found in 16S subunit, and the 18S ribosomal RNA is found in 14S subunit. 18S ribosomal RNA contain 2100 nucleotides, and 5S ribosomal RNA contain 120 nucleotides. 5.8S ribosomal RNA contain 160 nucleotides, and 28S ribosomal RNA contain 4700 nucleotides. 18S and 28S ribosomal RNA contain a characteristic number of methyl groups, mostly the 2'O methyl ribose. The molecule of 5S ribosomal RNA has a clover leaf shape and has length is equal to 120 nucleotides. The 5.8S ribosomal RNA is associated with the 28S ribosomal RNA molecule and therefore referred to as the 28S associated ribosomal RNA. At several points, the ribosomal RNA is highly looped, double-stranded, and helical, and at these regions, it bears a hairpin loops between the complementary regions of the same linear chain. The base pairing between these loops give a double helical configuration. The ribosomal RNA actively participate in protein synthesis. In bacteria, an interaction between the 16S ribosomal RNA and the mRNA is believed to help the smaller ribosomal subunit to recognize the starting end of the mRNA for binding to it. Ribosomal RNAs are also suspected to provide some catalytic activities for the protein synthesis. Next is the ribosomal proteins. The 17S ribosome of each E. coli is composed of 55 ribosomal protein. Out of these 55, 21 are from 13S subunit and 32 or 34 proteins are from the 15S ribosomal subunit. 18S ribosomes contain 17 to 18 kind of protein, and 14 to 15 proteins are from 16S subunit, and 13 to 14 proteins are in 14S subunit. And these ribosomal proteins serve as enzymes to catalyze the multiple reactions of the protein synthesis. Their energy source is GTP. They are found in four major groups: transverse factors, initiation factors, elongation factors, and the termination factors. Ribosomal RNA has a central role in the catalytic activities of ribosomes during the protein synthesis, and the ribosomal protein have found to be a major role in enhancing the catalytic functions of the ribosomal RNA. Next is the metallic ions. The major metallic components of the ions found in the ribosomes are the magnesium, calcium, and the manganese ions. The proteins and the ribosomal RNA get linked through the electrostatic forces and by the magnesium ion complexing. Electrostatic forces include the salt linkage between the phosphate groups of ribosomal RNA and the amino acid groups of amino acid residues of the proteins. Magnesium ion complexing involve the binding between the phosphate groups of ribosomal RNA due to an optimal concentration of magnesium ions. This is reconstitution experiment of ribosomes. This experiment is conducted by the Nomura and Thorpe to understand the three-dimensional organization of ribosomal proteins in the ribosomes, and also investigating the interaction between the molecules of ribosomal RNA. And this experiment involved taking a purified 13S ribosomal subunit and get dissociate into its components, that is ribosomal RNA and the proteins, by the chemical means, and then allow them to reassociate under the appropriate ionic conditions. The dissociation is brought about by the treatment with 4 molar urea and 2 molar lithium chloride, which separate the proteins. The reconstitution is take place in 2 steps. In the first step, at low temperature, 16S ribosomal RNA become bind to some of the 13S ribosomal proteins and forming a reconstitution intermediate particle, that is the RA particle. In the second step, RA particles are heated at 14 degrees Celsius in the presence of other proteins that were found in the supernatant, and they are forming an excited intermediate. Within 20 minutes, fully active 13S ribosomal subunits are formed. The self-assembly of 13S subunit is highly specific. It can be achieved with the 16S ribosomal RNA of the other bacteria, but not with the 16S RNA from yeast or the 23S RNA from the E. coli. In the similar manner, the reconstitution of 15S ribosomal subunit is achieved. Finally, a complete functional ribosomes is reconstituted spontaneously. And these are all about the reconstitution experiment of the ribosome. So in this video we have discussed about the structure, chemical composition, and the reconstitution experiment of the ribosomes. Thank you.